Hello, today we are going to learn about DNA sequencing. DNA sequencing is the process of determining the exact order of 4 bases, adenine, thymine, cytosine and guanine in a given DNA template. Knowledge of DNA sequences enables a greater understanding of the molecular basis of life, providing several information on a wide range of biological processes. Information on DNA sequences and the ability to manipulate these has accelerated the development of biotechnology and has led to the development of molecular techniques that provide the tools for asking and answering several important scientific questions. You have nearly 3 billion bases arranged in an order in your genome. They code for more than 20,000 genes to synthesize proteins. Obviously, humans or us have a larger genome. What about other organisms? Yes, they have different size genomes. But the thing you need to remember is genome size does not reflect its complexity. Let's talk about DNA sequencing, which is simply defined as the process to elucidate the order of bases in an organism's DNA strand. That means a total DNA strand. Since you know there are different sized genomes in the organisms, we can easily understand that one sequencing method may not be useful to sequence several genomes. As mentioned earlier, there are several reasons why do we need to sequence the genomes. Basically, order of bases or nucleotide sequences in your DNA affects your health. It gives you details about the diseases you might have or you are prone to, along with how you are going to react to certain kinds of medicines. Suppose you need to clone a gene. After the transformation process of the recombinant vector, it's very important to check the presence of the actual gene you need to transfer. For that, you need to sequence the gene to make sure you have done the thing right. Forensics, another interesting discipline that uses DNA sequencing. You have the biological evidence from the crime scene. Also, you have the suspects. All you need to do is compare the DNA sequences of those two kinds. Voila, you got the actual criminal. Let's have a look at the methods that we can use to sequence genomes. But we have to start from the beginning. In late 1970s, researchers tried to develop several methods to sequence purified DNA. Pioneers are Maxam Gilbert and Frederick Sanger. First, let's talk about the Maxam-Gilbert method, the chemical cleavage sequencing. First, you need to get the sample DNA and make it single-stranded with the addition of a label at one end. Here, we are adding a P32 molecule and labeling in the 5' end of the DNA strand that we need to sequence. Then, the DNA strand is exposed to a base-specific damage, or the DNA strand is cleaved at specific bases with the use of several chemicals. Here, we use 4 different chemicals. For example, in mixture number 1, we are adding dimethyl sulfate. That mixture specifically cut the DNA strand at guanine base. Like that, in mixture number 2, we are adding formic acid. It can cut at both adenine and guanine bases. Likewise, there are 4 different chemicals using in this process. Reaction is controlled, so the cut happens only once. Obviously, here we don't know the DNA sequence, but for example, we'll think it's the sequence given above. If you get this sequence, you can see at the 4th place, there is a guanine nucleotide, guanine base, and also in the 10th place. In all these mixtures, there is a possibility to have no cleavage at all. So you can see in all these mixtures, the first line, the base sequence in the first line contain almost the same as the one in the DNA strand. So that means there is no cleavage at all. We have the total 12 nucleotides. Let's look at the first after that. In mixture number 1, there are 2 places of guanine. So as a result, there can be 2 cuts. So you can get 2 strands like this. We can observe the strand to left because we have labeled the 5' end. Likewise, in mixture number 2, you will cut at either adenine or guanine base. When cut at adenine base, since there are 2 adenines in the middle, that can result 2 strands, as you can see. So as in the mixture number 1, this also cuts at guanine. So as a result, again, there can be 2 other strands. Similarly, there will be several strands in all mixtures, as you can see in this picture. Now you can read the gel to score the bands. There is a special method to do that. Bands appearing in 1 and 4th lane, that means G and C lanes, are read as G and C directly. That means as guanine and cytosine bases directly. But in both A and G lanes are taken as guanine. The bands in both these lanes are taken as guanine. But the bands that are only in A plus G lane are taken as adenine. Similarly, bands appear in both T plus C and C lanes are taken as cytosine. And bands that are appearing only in T plus C lane are taken as thymine. Now, let's read the sequence, starting from the top. Larger products tend to stay at the top, as you know. So we can start here. It contains all the nucleotides, but we don't know that. In the 12th place, here you can see all the bands having all nucleotides. Let's look at this place. It's a band in T plus C lane, and you can see the same band in C lane too. So as we discussed earlier, since the band is appearing in both these lanes, we are taking that as a C. So the thing is same for the next band also. Since that also appearing in both T and C lanes, we are taking that also as a C. Let's look at this band. That is there in A plus G lane, and also we can see that in the G lane. So therefore, we are considering that as a G. Then what about this one? That is only there in the A plus G lane, but that is not there in the G lane. So what is that? Yes, we are taking that as an adenine. Likewise, we can read the bands in whole gel. Finally, the sequence has to be read from the bottom of the gel to top. There, now you have the sequence of the unknown DNA strand. This was a simple method. However, this was not a very successful one. The data produced by the method are considered ambiguous. That means there can be several ways for us to interpret them. Also, multiple hazardous chemicals and radioactive materials are used, which is not good. Especially, this method can be used only to sequence a short range of DNA strands. That means only up to 500 bases. Therefore, they had to go for another method, which is Sanger sequencing or dideoxysequencing method, the one that became most widely used. Let's learn the Sanger sequencing method in our next video. Hope now you have a good understanding about the Maxam-Gilbert method of DNA sequencing. Have a good day. Thank you.